Hello, we're here with John Oberdick from
Hi.
the Ohio State University. Could you please tell us a little bit about yourself and your academic career, your current projects?
Sure. Do you want to know where I went to graduate school and all
For
that?
example, yeah.
So I got my PhD at Columbia University in biology department, and I worked with Cyrus Leventhal, who actually did his postdoctoral work with Jacob and Monod in Paris. And he had a number of different projects in the lab, one working on protein folding, that was the biochemistry part. And then the other one was working on neuroscience, which was using zebrafish to reconstruct the visual system. And so he was sort of a pioneer on computer modeling for both protein folding and then trying to design, or try to figure out the wiring of the nervous system, but using computer models to do three-dimensional reconstructions, that kind of thing. So that was partly his physics background, but this whole project that I was working on led to trying to identify markers in the cerebellum that identified unique regions of the cerebellum, because extending the work from the visual system, the optic nerve projects to discrete areas of the tectum. So it's one of the classic systems for looking at axonal pathfinding and targeting. The cerebellar system was thought to have a similar precision in terms of the wiring. And so, since it was a system that was, there's a lot of mutants that were just naturally available from various mouse colonies, classical mutants known as Staggerer, Lurcher, Weaver, etc., etc. At that time, most of the genetics having to do with a neuronal system seemed to be involving the cerebellum. So we decided to use the cerebellum to work on cloning cDNAs that were specific, cloning cDNA molecules that were copies of RNAs from the cerebellum, using some tricks to identify some of those cDNAs that might be specific markers for Purkinje cells. And so anyway, that was sort of the history of that. And actually out of that PhD project came the gene L7, which a lot of the talks at this symposium have talked about. And I continued that work as a postdoc. So I crossed the river from New York City to do a postdoc over at a place called the Roche Institute of Molecular Biology. And so as a postdoc with Jim Morgan, who was a professor at the Roche Institute, I cloned the mouse gene that encodes the L7 mRNA, and that's when we obtained the promoter, and we were able to show that promoter, when linked to other coding sequences for other proteins, could be used to express foreign proteins in mice just in Purkinje cells. So that all started in my postdoctoral work, but all of that was influenced by my PhD work. And then my postdoc finished in 1992, and then I moved to Ohio State to take on my first faculty position.
Wow,
and
so actually you're quite a long time already there.
Yeah, where? At
the Ohio State University.
Oh yeah, I've been there since '92, so it's going on 25 years.
Yeah.
So it's been, it's been a great place, and I decided to go there actually because there was a number of cerebellar researchers there. My chairman of my department when I first started there was James King. He's a well-known cerebellar anatomist. So of the places where I got offers to go, I chose there because the research resources was one thing. The amount of resources, and they had a transgenic facility that was just being built, and so sort of state-of-the-art facilities. But having a chairman who was actually a cerebellar biologist was another reason. So that Yeah, so that's sort of the short. What else do you want to know? What's-
Yes, another question would be, how did you come to be at the seminar today?
Oh, well, Yaroslav invited me.
Yeah, but do you know each other from?
Oh, well,
Past,
the reason we know each other is because Yaroslav is one of the people who has capitalized on the use of the L7 promoter, which again goes back to the Cyrus Leventhal laboratory at Columbia, and he was one of the people who. So he created the L7 Cree mouse, which I would say tens, probably hundreds of investigators have used to do specific Purkinje cell specific knockouts. So I guess I don't think I've ever met Yaroslav before, but our research pathways are certainly linked together because of the discovery. I guess I discovered the L7 gene. He created the L7 Cree mouse, and so we're linked in that way.
Okay, great. Thank you very much.
Sure.
And the last question I have for you is kind of a funny one. If you could choose to be any species you want, what species would you choose and why?
Well, I don't know. I've made so much of my life working with mice, but I don't know. I guess not. I guess I wouldn't want to be a mouse.
Or maybe just an animal ability that you'd like to have,
Animal ability.
like flying or diving very long or something. You like the way you are.
I do like the way I am, but I'm trying to think if there is any characteristic that's already intrinsically part of me that's animal-like, or any particular animal. But I guess from the talk I heard today, I tend to be independent. I like to be alone a lot. So I hear, according to the talk today, bears are sort of, after they leave their mother, they tend to live alone. So bears or wolves maybe, because I get to range far away and be on my own and independent. Maybe
And also the beautiful landscape.
Yes,
Where
right.
bears and wolves
Yeah.
live in. Yeah.
So maybe the independence would lead me to be a bear or a wolf or a...
Okay, great. Thank you very much.
Sure.
And have a great evening today and
Great,
great
I'm
time
having,
here in Poland.
Oh, I intend to have a good time.
Okay, great.